So the next speaker right now is going to be Jessica Lau from Dr. Tuzinski's lab, and she's going to be talking about a regulator of the DNA damage checkpoint.
Thank you, Oliver. Good morning, everyone. Okay, let's see. All right. Good morning, everyone. So I'm Jessica Lau. I'm Dave Tuzinski's lab. I'd like to start off by thanking the organizer for giving me an opportunity to share my work with everyone. So today, I'm going to tell you a little piece of a puzzle from a proteomics screen that I did. In particular, how the DNA damage checkpoint targets the exoribonuclease, Xrn1, in response to damage. So DNA in our cells is prone to damage, and it must be repaired in order to maintain genomic stability. So the DNA damage response, or checkpoint, is highly conserved. The sensor kinases, MEK1 and TAL1, are recruited to sites of DNA damage and are activated there. The signal is then amplified to Rad53, the effector kinase, and then Rad53 go on to regulate a variety of cellular responses, including regulation of transcription translation, DNA repair, and delaying the cell cycle. Evidence in the literature has suggested that the DNA damage response also regulate other pathways in cellular metabolism. Some of this work was prompted by the work of Audrey Gash in early 2000, where she did a transcriptomic analysis of, in response to DNA damage. And so my goal was to broaden the understanding of the DNA damage checkpoint by identifying targets in pathways that have not been previously implicated in the DNA damage response. And to do this, we did a proteomic screens for Rad53 targets. So Rad53 is not known to have sequence specificity in its phosphorylation sites. And targets of Rad53 can generate a laddering effect or a smear in response to DNA damage on an SES page gel. So you can imagine your favorite protein here in orange. It has a solid band in normal condition. In response to DNA damage, you see laddering effects. This could be a result of phosphorylations on the proteins at multiple sites. And depending on the number of phosphorylation sites on that target, it might run differently on a gels. So this indicates that there could be multiple modified species. So our approach was to overexpress Rad53. And we reasoned that if we can saturate the phosphorylation sites on the substrates, we would be more likely to detect that modified substrate. We also deleted phosphatases that are known to counter the effect of Rad53 in order to stabilize the phosphorylated residues. So let's see how this works in the cell. Here. So you can see in wild-type cells, in response to DNA damage, you see a shift, a modification of Rad53 targets, SLID3, and NDD1 here. And you can also see that MEK1 TAL1 substrate is also phosphorylated and damaged. In the triple phosphatase deletes, you can see modification of Rad53 substrates as well here, but only in the presence of DNA damage, indicating that deletion of these phosphatases alone do not enhance the activity of Rad53. And now this is a strain where we overexpress Rad53 by driving its strong expression from a galactose promoter. And you can see here that in the presence of galactose, where Rad53 is overexpressed, even in the absence of DNA damage, you get a very strong modification of these Rad53 substrates. This indicates that when you overexpress Rad53, it also activates it. Additional DNA damage in these strains does not enhance the modification much, so there's only a moderate effect there. There you go. You can see that activation of Rad53 alone is still specifically modifying Rad53 target. So therefore, overexpression of Rad53 activates this checkpoint in the absence of damage, but Rad53 retains its substrate specificity. So here in our strain, we've deleted the phosphatases that counter the effect of Rad53. In one strain, we also deleted Rad53. In the other strain, we are overexpressing Rad53 and as well as the downstream kinase DUN1. Induce DNA damage, we extract the proteins, trypsinize it to cut the proteins up into small pieces, enrich for phosphopeptides, and perform a mass spec analysis. This was done in collaboration with the Kogan lab at UCSF. And we looked for an enrichment of phosphopeptides in the strain that's overexpressing the kinases. We found over 40 proteins where there are greater than a 25-fold enrichment in the Rad53 strains. I'll go over these, some of the features of these with you in the next few slides. I also like to point out the size of the bubble corresponds with their fold enrichment. And you can see here that Xrn1 belongs to the class where it's, the phosphopeptides are greatly enriched. And in fact, Xrn1 is our best hit. It's over 200-fold enriched in the kinase expressing strains. Oh, sorry. And so we found many of the substrates were not previously identified as Rad53 substrate, which is great, but we also found substrates that were previously known or previously identified, and that's very reassuring. So next we wanted to validate these hits. So we used two approaches. One was to look at the mobility shift, and we found that 17 of the 40 here in yellow produced a DNA damage dependent mobility shift. But not all substrates that are modified produce a detectable shift on a gel. So I also wanted to confirm whether they can be directly phosphorylated by Rad53 using an in vitro kinase assay. And I confirmed that half of these substrates can be directly phosphorylated by Rad53 in vitro. Comparing the two, we see that about a quarter of these hits are validated by both assays, but 3/4 of them can be validated by at least one of the two assays. And as you can see here, Xrn1 is validated by both the mobility shift assay and the in vitro kinase assay. And I will show you those data later in the talk. There's an interesting pattern that we also observed in the data set, and that was the number of hits that were involved in RNA biology, shown here in red. We can further break this down into hits that are involved in mRNA biology in purple, or rRNA biology in orange here. For the rest of this talk, I will focus on Xrn1. So Xrn1 is a very conserved 5' exoribonuclease. You can see the nuclease domain is conserved in all species of Xrn1, and there's also another conserved region that is required for the nuclease function. And much of Xrn1 was characterized by Wolf-Heyer in the 90s. Xrn1 is involved in all aspects of RNA biology. It is not an essential protein, but the mutant has a lot of variety of cellular defect. Most interestingly, it is sensitive to DNA damaging agents, indicating that it is involved in the response to DNA damage. Xrn1 is the main cytoplasmic mRNA degradation factor. Its preferred substrate is a 5' monophosphate mRNA, which can be generated by decapping of mRNAs or endonucleolytic cleavage of that mRNA. So here you have a transcript, which is stabilized by the presence of a 5' cap and a poly A tail. This can be degraded in several ways. First, the poly A tail can be deadenylated, followed by decapping. Decapping can occur in the absence of deadenylation, and also this mRNA can be cleaved internally by an endonucleases. In all cases, you form these 5' monophosphate mRNAs that is a substrate for Xrn1, and the 3' end mRNA can then be cleaved by the exosome. Additionally, we found several factors that came out in our hits that are involved in this pathway. So there's PAB1 that binds poly A tails, DCP2 is required for decapping, and SCI7 is part of the cytoplasmic exosome complex. This indicates that this pathway is likely regulated by the checkpoint in response to damage, given the number of hits that came out. So here we confirm the Xrn1 shifts in response to DNA damage. So you can hear in wild-type cells, you can see a shift there. This is dependent on Rad53. As Rad53 deletion, you abolish the shift. Then we also want to see if it requires the downstream kinase of Rad53 den1. And here we can see that this Xrn1 still shifts in response to DNA damage in a den1 delete, so it is den1 independent. So we can place Xrn1 downstream of Rad53. Now we wanted to see if Xrn1 can be directly phosphorylated by Rad53, if it's a direct target. So to do that, we did the in vitro kinase assay. So we purified Xrn1 as well as active Rad53. Then we looked for phosphorylation by p32 incorporation. So the top panel is an auto-rad showing p32 signal. The bottom panel is a Western of the GFP tag substrate. SML1 is the den1 target, And you can see that in the presence of DEN1, you see a very strong p32 signal where SML1 runs. Here's Xrn1. You see that in the presence of Rad53, you see a very strong p32 signal where Xrn1 runs. And this is specific to Rad53, as in the presence of DEN1, you do not see any signal there. So this indicates that Xrn1 is a Rad53 specific substrate. These are negative control, and you see very little p32 signal in any of the negative control, indicating that Rad53 and DEN1 are not promiscuous kinases in vitro. So now what we really want to know is what are the biological consequences of Xrn1 phosphorylation by the checkpoint. We tested a few candidate-based approaches, and those have not been very revealing. So since then, we've conducted an unbiased screen for suppressors of Xrn1 damage sensitivity. So you can see here, these are spot tests in the absence of damage or in the presence of damage. And you can see both the slow growth defect of Xrn1 as well as its extreme damage sensitivity. So we've identified a lot of suppressors. So this is just a partial list of some of the suppressors that we found. And you can see that we've identified suppressors that both rescues the slow growth defects of Xrn1, such as suppressor 3 and suppressor 6, as well as suppressors that also suppress the damage specificity of Xrn1 only, such as suppressor 4 and suppressor 8. We've also identified that a few of these are dominant suppressors, but the majority of them are recessive suppressors. And we're currently working to sequence these strains to determine the identity of the suppressors. I would like to end with a model for how we think that Rad53 might be regulating Xrn1. We've mapped the phosphocytes on Xrn1, and most of it maps to the C-terminal of Xrn1, which is a very large protein. So what we think is what I want to add is, so given that Xrn1 is sensitive to DNA damage, we think that it's doing something positive during damage. So therefore, the phosphorylation of Xrn1 by Rad53 is likely to be an activating regulation. So we propose that the DNA damage checkpoint might be enhancing the activity of Xrn1 during the DNA damage response, so such that in the absence of damage, Xrn1 retains its basal activity to carry out the necessary functions of mRNA decay. In the presence of DNA damage, we think there's a checkpoint-dependent enhancement of Xrn1's activity, such that now you have increased RNA degradation, and this may help to reduce levels of some RNA species that might be deleterious during DNA damage. And in the absence of the checkpoint, Xrn1 retains the basal activity, which is not sufficient to reduce those levels of RNA species, and you have an overabundance of deleterious RNA species. So we are currently working to test this model. And we have a lot of experiments to do. And with that, I would like to thank everyone for listening, also the T-Lab, in particular my mentor, Dave Tuchisky, as well as Katie and Leonie, who have helped me directly with the project. I also would like to thank our mass spec collaborators, Nevin Krogan at UCSF and James Walsh-Hagel at UCLA, as well as Damon Runyon for funding. Thank you, everyone.
Bit of a technical question about your screen. Presumably when you're overexpressing Rad53 with GAL, it's sort of pan cell cycle. Did you make any attempt to limit it to S phase or G2? Actually,
no, we didn't. Yeah, we thought about doing that, but when we did the screen, we weren't interested in just cell cycle specific, so we started off with just asynchronous cells. But yeah.
Hi. A number of the RNA binding proteins that you found are in stress granules or X bodies, I think. Does Rad53 affect localization?
So we looked at Xrn1 localization and stress, and Rad53 doesn't appear to affect its localization.
Nice story. So the other thing I noticed that when the XRN gets activated, there's a lot more ribonucleotides that are made. Do you think that that has any effect on the cell so that the pools of deoxynucleotides can go up as well, because they are that's the rate limiting step, you know?
Yeah, possibly. Well, the way I've drawn it, there's more DMP because, or RNP because it's been more degradation, right? I'm not sure if that affects it, but perhaps. I mean, there's nucleotide metabolism and DNA damage response is very connected. And I think there's data in the literature to look at, to suggest that Xrn1 delete might have effects on the levels of RNR transcripts, so there could be some connection there.
Thank you very much.
Thank you.